Because IBD most heavily impacts the intestines, it is best to start there. The intestines are a long, continuous tube running from the stomach to the anus. The intestines are necessary to absorb the nutrition in food, provide a barrier against bacteria, and produce immunity against bacteria. IBD are inflammatory bowel diseases in which the intestinal lining or mucosa is inflamed. This disease impacts 1 in every 250 people, which translates to about 1.4 million Americans. IBD is mainly split into Crohn's disease and ulcerative colitis, which affect the small or large intestine and colon or rectum, respectively. The symptoms of IBD include, but are not limited to, eye inflammation, stomatitis or mouth ulcers, skin ulcers and sores, liver and bile duct inflammation, vomiting, diarrhea, abdominal pain, cramping, ulcers in the digestive tract, rectal bleeding, and joint pain and swelling. Scientists don't know the exact cause of IBD, but they've correlated it to genetic and environmental factors. So how does altered cell signaling cause IBD? Well, first we have to understand the intestinal mucosa's role with environmental pathogens. The first part of the mucosa is the intestinal epithelial cells, which are drawn in black. Another part is the mucus, which is shown in blue, and bacteria, which is shown in purple, is in the mucus. Two types of intestinal epithelial cells are Paneth cells and goblet cells. The intestinal mucosa serves as a barrier in at least 3 ways. First, the tight junctions prevent bacteria from entering. These are drawn in silver. Also, goblet cells secrete mucus and Paneth cells secrete defensins. Tight junctions and mucus prevent bacteria from entering, and the defensins destroy bacteria by a process called cell lysis, which disrupts the cell membrane. The Paneth cells also communicate the gut's bacteria levels with other cells called macrophages in order to help the immune system with bacterial immunity. This works by a typical receptor signal transduction pathway. Here's a closer look at this Paneth cell. Paneth cells have a receptor protein called toll-like receptors, or TLR for short. When a bacteria, which is shown in purple, binds to the TLR, a protein known as MID88 that is connected to the TLR is activated and the reception turns to transduction. The MID88 protein binds to a card15 protein, which is being colored yellow. This leads to card15 protein inhibiting a protein complex with IK-beta bound to P50 and RelA. This inhibition removes IK-beta, leaving a new complex with just the P50 and RelA protein called NF-kappa-beta. The special aspect about the NF-kappa-beta complex is that it's small enough to enter the nucleus, which has a C in it. In the nucleus, the NF-kappa-beta complex transcribes the DNA, or activates genes. This leads to the production of the TNF-alpha protein. As a review, the reception occurs when a bacteria binds to the toll-like receptor. The transduction is the production and journey of the NF-kappa-beta complex, and the response is the TNF-alpha protein to be produced and released. Here is a graphic of the TNF-alpha protein. When it is released from the Paneth cell, it binds to a receptor protein on the macrophage cell, calling for an appropriate immune response to the bacterial level. In IBD, the signal transduction pathway is almost exactly the same. In one explanation, the card15 protein is mutated, thus amplifying its signal. This amplification leads to increased NF-kappa-beta production, meaning more DNA transcription, finally facilitating increased TNF-alpha production. So, more TNF-alpha is released from the Paneth cell. Because the function of TNF-alpha is to indicate bacterial levels to the immune system, its increased production indicates to the body that there is more bacteria than there actually is. Consequently, the immune system builds up its attacks, but there is no bacteria to destroy. This means that the body attacks itself, and specifically at the site of the intestines. This attack expresses itself in several ways, including angiogenesis, or blood vessel growth, Paneth cell necrosis, or death, intestinal epithelial cell death, and finally, an immune response resulting in even more TNF-alpha production, making IBD a chronic disease. The intestinal mucosa in IBD is deteriorated. Bacteria can infect the lumen due to the lack of the epithelial barrier, nutrition can't be absorbed, leading to extreme diarrhea and weight loss, and TNF-alpha continues to be produced from further immune response. This leads to inflammation in the form of swelling and damage. Here is a colonoscopy view of a normal, healthy intestinal mucosa. And here is the broken down intestinal mucosa of a patient with IBD. Although IBD is incurable, there are many treatments. One is called infliximab, and it targets the TNF alpha response by simply eliminating TNF alpha. At a more microscopic level, this anti-TNF antibody, which has a human part with the information shown in pink and a mouse part to attack, shown in silver, binds to TNF, neutralizing it. With the neutralization of TNF alpha, the intestinal mucosa can heal, effectively treating IBD. Does infliximab sound too good to be true? It might be. Infliximab has some scary side effects. TNF alpha levels may drop too low, making the recipient prone to infections and sometimes cancer. For now, IBD doctors have to keep in mind that IBD is a chronic disease and must balance symptoms and treatment. Hopefully, one day we'll find a cure.